So like I said, guys, it doesn't show your images on it. It's just me. So yeah, if we just have a go over these questions, and we'll give 1 more minute, see if, oh yeah, Salma's just joined, see if Sarah's joining as well. Then we'll have a go. Hi Salma, you okay? We're just having 10 minutes at the start. We're just going to go through these questions. So this is just, you know, a bit of revision from last week, what we did about stem cells. So just how to go. I know I think you had a test, did you? So I hope that went well for you. Right, just do 1 more minute and then we'll start going through, all right? Ooh. Oh, that's great, Salma. I'm sure it went very well. Okay. So does anyone want to start with the first one? So if we're not sure, doesn't matter. So you can send me in the chat, or you could just say out loud. So how do totipotent stem cells differ from pluripotent stem cells? Do we remember? Just give you a second if anyone wants to send an answer. Okay, perfect. So Zarwin, I think that one, if you meant to say can, isn't it? You've put can't here, but great. So Zarwin, that's a really good answer. And Rhea, you've put, yeah, great. So everyone's giving me a really good answer there. Okay, so I'll just read out Zarwin's because everyone's done the right answer here. So Zarwin's put totipotent can differentiate into any cell. Okay, pluripotent can't differentiate into placental cells. Yeah, perfect. So you're all right. And everyone's just put a variation of that. So remember, totipotent can differentiate into any type of cell, including the placenta. Pluripotent, any type of embryonic cell, any type of body cell, not the placental cell. And then multipotent are only specific types of cell. So like I said, we used the example of in your bone marrow can differentiate into different types of blood cells. So this is a really easy one. Second one, give 2 examples of specialized cells in a human. So just any 2 will be great. Great, some good examples here, guys. Just wait for everyone to answer. Oh, Sarah's just joined. That's great, well, Ada. All right, perfect. So I've got a couple here. So I don't know if you're still sending one, Salma. But so we've got blood cell, nerve cell. Yeah, Salma, you put nerve cell as well. We've got root hair cell, sperm cell. So, hey, Sarah, we're just doing some questions. I don't think you were here last week, but we're just doing some questions on stem cells, just a bit of a recap. Okay, don't know if you got to watch the recording or anything like that. So we're just doing a couple. So the first question, how do totipotent stem cells differ from pluripotent? Totipotent can differentiate into any type of cell, okay, including placental cells in the placenta, but pluripotent can only differentiate into a body cell, so it can't differentiate into a placental cell, but it can still, you know, differentiate into lots of different types. And then our second one here, we're just on number 2, so we just had some examples. So people have put root hair cell, sperm cell, blood, and nerve cell. I think of some more muscle cell. Oh, we've already done a blood cell, so red blood cells or white blood cells. So there's lots of different examples. Okay, right, let's move on to number 3. So some stem cells can be found in adults. Over the past few decades, more research has been performed on adult stem cells and their properties. So leukemia is a type of blood cancer. Explain how cells found in adult bone marrow can be used to treat leukemia. Okay, can anyone remember the 2 steps that we use to treat leukemia? Great, so Zareen, so you've got here stem cell transplant. Zareen, can you remember how we do that? If you want to unmute, can you remember?
Do you basically have to replace the healthy stem cells in the bone marrow and replace the unhealthy ones with the healthy ones?
Perfect. So do you remember, so Salma, you put great here, so chemotherapy. So we're going to have to get rid of all of our unhealthy stem cells, aren't we? Because remember, those unhealthy stem cells are differentiating into lots of different types of blood cells, but they're all cancerous and unhealthy. Okay, so we need to kill someone's stem cells that they've got in their bone marrow. And then, like Zareen said, a stem cell transplant. So let's say Zareen's given me a stem cell transplant. Take the stem cells out of the bone marrow in Zareen's inside the bone and give it to me. Okay, and then I start to use Zareen's bone marrow and stem cells to make my own blood type. Okay, so a cool interesting fact is, let's say Zareen's got type A blood and I've got type O. If I get a stem cell transplant for Zareen, I'm going to get type A blood now. Okay, just because I've got Zareen stem cells. All right, so just a little fact there. Okay, so that's how we treat that type of blood cancer. So firstly, we need to kill the cancerous blood cells, as cancerous stem cells. Then we need to do a stem cell transplant, so the person with cancer can start making healthy blood cells. Okay, perfect, guys. And last one here, discuss the scientific advantages of using adult and embryonic stem cells. Okay, so let's split this answer up into 2 because it's discuss. Okay, so it's just the advantages, so you don't have to talk about the disadvantages here, but let's split it up into adult and then let's split it up into embryonic. Okay, so what are the advantages of adult and then what are the advantages of the embryo stem cells? And Sarah, don't worry if you going over your head because you went in that lesson. You could maybe have to listen to the recording because we went over it. Okay. So Zareen, you've put here, so you can discover how to treat a certain disease. Yeah, great. So great, Ria, this is like the main one here. So adult is, so you put unethical, but adults is more ethical, isn't it? Because you're right, people usually volunteer to donate the cells, so they give their consent. Okay. And yeah, so Zareen, so adult are more specialized, so that would be kind of a slight disadvantage, wasn't it, of the adults? Okay, so let's do embryonic. So what are the advantages of using the embryonic stem cells as opposed to the adult? Someone, yeah, Salma, great. So can differentiate to any type of cell. Okay, so remember our 3 types of stem cells: totipotent, pluripotent, and multipotent. Okay, yeah, perfect, Ria. So you put embryonic as totipotent. Okay, so we get our totipotent and our pluripotent stem cells from an embryo, and it depends how much the embryo is developed. Okay, if it's not developed very much, it's going to be totipotent, isn't it? But if it's developed a little bit, then we're going to get pluripotent stem cells. Okay, so they can differentiate to any type of cell at all. Okay, but I know this question is just advantage, but the disadvantage of that is the advantage of the human ones, isn't it? It's not very ethical. You know, that embryo could develop into a baby later on. Okay, so people argue. All right, and also that embryo can't give consent to its cell being used, and you don't know what's going on. Okay. So advantages of using adult is that you can get it from a living donor, they can give consent, okay? And it's especially good, what we just talked about in bone marrow transplant. But the advantage using embryonic is it can differentiate into any type of cell, all right? So that's a little bit better. Okay, great, well done, guys. So you seem to remember a lot from last week, which is Really, really good to hear. All right, so we've probably just got one lesson on transporting animals left. Okay, so let's try and finish that off. So at the end of last lesson, we were just starting to go through the graph which showed pressures. Okay, so let's try and get through that, because it's a really important graph that you need to kind of get your head around. So we might revisit it at another time. Okay, so this is the one here. Let me just do this. So remember, we were saying that we were just going to do the left side of the heart. So let's just draw out one side of the heart. Okay, so this is going to be our left atrium, and I'm just drawing one side of the heart. Okay, let's draw our AV valve. Okay, our left ventricle, and then let's go up to the aorta here, and then this is our semilunar valve. Okay, so this is what we're doing here. So we had done the very start of this graph, but let's go over it again because Sarah wasn't in the lesson as well. Okay, so if I get my little laser so you know where I'm talking about on the graph. So let's have a look. So remember, the blue line was the atrium. All right, so the smaller little bit at the top of the heart. Okay, and the green line was the ventricle. Okay, which is this bit here. And then the red line was the aorta, so the big blood vessel, the big artery coming out of the heart. Okay, so you can see at the bottom here that this has been split up into 3 sections. Okay, so this first section is when the atria are contracting. So we've got to remember this is happening simultaneously on both the left and the right side of the heart. But just for simplicity, we're just going to talk about the left. Okay, if we were talking about the right, this red one, this would be the pulmonary arteries, so the aorta. Okay, and the pressure would probably be a little bit lower. All right, but let's just talk about the left for simplicity. Okay, so the atria are contracting in this little bit, so this little chamber. Okay, that means the pressure inside the atrium is higher than the pressure inside the ventricle. You can see that here, can't you, in this first section? Okay, so blood is going to go from the atrium here into the ventricle. Okay, see that happening there. I've drawn the valve the wrong way around. I've just seen. Okay, doesn't matter. So blood's going to flow from the atrium to the ventricle, because like anything, we go from high pressure to low pressure. That's where things naturally want to go. When you think about diffusion, that's high concentration to low concentration. Okay, so we start to do that. Okay, and this is what we call atrial systole. So I can't remember if I mentioned that last time, but let's write that here. Okay, there we go. Atrial systole or systole. People say it different ways, okay? And that just means the atria is contracting. Okay. So when we get to the end of the atria contracting, the pressure in the atria starts to decrease, okay? And the ventricles start to contract, all right? So this next stage is called ventricular systole. Okay, so this next stage. But you can see here that the lines cross, okay? So the pressure in the ventricles starts to exceed the pressure that's in the atria, all right? So what we know about the movement of blood, it wants to go from high to low pressure. So now in the heart here, we've got blood in the ventricle, all right? So the blood's going to want to start going from high to low pressure this way, okay? But we don't want that to happen. So what happens here is we've got this blood. This valve here, our AV valve, closes. It stops the backflow of blood, okay? So at this point on our graph, the AV valve closes, all right? So when we're looking at this graph, it's really important to know which valve we're talking about when the lines cross, and does it open or does it close? Okay, so it might be a good idea to screenshot this at the end or copy this diagram down. Okay, so the AV valve closes, okay, at this point, and that's to stop that back pressure going on. Okay, great. So our AV valve has closed. All right, stops blood going back into the atrium. All right. As the ventricle continues to contract, the pressure rises up and up and up, and it starts to exceed the pressure of the aorta, so the big blood vessel. Okay, so now blood is going to want to go from high to low pressure. So it's want to go from the atrium into the aorta. Okay, so a little diagram at the side. So now that little valve is going to open. And remember, that's our semilunar valve. Okay, so you can just put SL, valve opens. Okay, sorry, I'm writing on my laptop here, so it's a little bit messy. Okay, so opens. All right, and it's all to do with the pressure changes. Okay, so now the ventricle continues to contract. All right, continues, and it reaches a peak. Okay, it starts to come down slightly, and you can see here at the bottom, ventricular systole stops about here. So just when the ventricle pressure is starting to decrease slightly. Okay, and now the ventricle starts to relax. All right, so our next stage that we've got down here, our third stage, is called ventricular diastole. Okay, or diastole, people call it. Right, and that's how I spell it at the bottom there. I've just written it for you guys. Okay, so that just means the ventricle's relaxed. All right, so let's just go back. So we've got ventricular systole, and then the ventricle starts to relax. So the pressure starts to decrease. Again, it crosses over the pressure in the aorta. Okay, so now the pressure in the aorta is higher than the pressure in the ventricle. All right, so if we look at our little heart diagram at the side here again, blood is going to want to go from high to low pressure. It's going to want to go from the aorta back into the ventricle. So it's want to do this at the side here. We don't want that to happen. We need that blood to pump around the body. Okay, so what happens again is the semilunar valve closes. All right, to stop that backflow of blood. So here, where it crosses over, the semilunar valve closes. Okay, so I'll just put SL close, but it's semilunar valve. And again, remember, this is happening on the right side of the heart as well. Okay, great. So as the ventricle's relaxing, the pressure is going to rapidly decrease in the ventricle. Okay, so we keep going down. Keep going down. And now we get where it goes lower than the pressure in the atrium again. So here, so now blood is going to want to flow from higher pressure, which is the pressure in the atrium is higher, to lower pressure, which is the pressure in the ventricle. So now blood is going to flow from high to low pressure. So what's going to happen? That little bit, yeah, that valve is going to open. Okay, and remember, that's our AV valve, our atrioventricular valve. All right, so AV valve opens. Okay, great. So this is what you need to label on this diagram. You need to kind of understand about the pressures and do that. All right, once this happens again, ventricular diastole, okay, and then it just restarts again. We go to atrial systole, ventricular systole, and ventricular diastole. Okay, so you know those lub-dub we've talked about, those heart sounds. Okay, that's all to do with these valves opening and closing. Okay, they make a sound when they open and close, and that's what you can hear with your heartbeat. All right, so let's go through again, and then if anyone's got any questions, just speak up. Okay, so we start at the heart. Okay, the atria are contracting, okay? So they're decreasing the volume within the heart, so they're going to increase the pressure. The pressure inside the atria is higher than the pressure inside the ventricle. So if we look at our diagram of the heart, blood is going to flow from our atria into our ventricle, all right? Now, as the atria start to relax and the ventricles start to contract, the pressure in the atria is lower than the pressure in the ventricle. Ventricles contract, so now blood's going to want to flow from high to low pressure backwards, but that AV valve stops it, the backflow. So the AV valve closes, all right? The ventricles continue to contract, keep going up, and it exceeds the pressure that's in the aorta, okay? So now blood flows from the ventricle through the semilunar valve into the aorta. So that opens that semilunar valve, okay? Ventricles continue to contract, continue, reach a peak, and about here they start to relax, ventricular diastole, okay? The pressure in the ventricles goes lower than the pressure in the atria, so blood is going to want to flow from high pressure in the atria to low pressure in the ventricle. So it wants to flow backwards, okay? We don't want that to happen. We've got a valve there to stop the backflow. So the semilunar valve closes, all right? The ventricle's relaxing, keeps relaxing, the pressure's falling, and it goes lower than the pressure in the atria, okay? Now, the blood is going to flow from high to low pressure, so it's going to flow again from the atria into the ventricle. So even though the atria is not contracting, because the pressure in the atria is higher than the pressure in the ventricle, blood's going to just passively go in, okay? So it's not an active process at this point. When the atria is contracting, that's an active process, but not during the resting phase or the diastole, okay? So because blood's going to flow from the atria to the ventricle, the AV valve opens again, okay? Thumbs up or thumbs down if that makes sense, guys. And has anyone got any questions before we move on? Is that okay? So I can see quite lots of thumbs up there. All right. If we need to come back to it at any point, and what we'll do, not tonight, because we won't have time to do exam questions, I don't think anymore for this one. Okay, but we'll do some at the start of next lesson, okay? Remember that, guys, it's all to do with the pressure and it's all to do with the valve. Okay, and now my thumbs up. Great. So what's next about the heart? There's a little bit more about the heart that we need to know. Okay, let me just clear all these drawings. Oh, I've also put at the bottom here, you might get given this in the exam, you might not, okay? It's a common exam question. Cardiac output equals heart rate times stroke volume. You might get, you know, a couple of figures and you have to work out their stroke volume or you have to work out their cardiac output, okay? So cardiac output equals heart rate, which is the number of beats per minute, times stroke volume, which is the amount of blood that is pumped out in one heartbeat, okay? So basically it's just how much blood is pumped out of the heart in 1 minute. That's what cardiac output is, all right? Again, I'll try and get some questions for next week, okay? So this is our last little bit about the heart that we need to know about. Okay, and sometimes it can be, you know, a little bit of more complicated bit, okay? That's fine. So units for cardiac output. Oh, let's go back. So it's going to be, you know, the volume per minute, okay, whatever. So it's normally mil. Okay, so mil per minute, that's what cardiac output is going to be, okay. All right, great. So this is our last little bit we need to know about the heart, okay. And what I'm going to do, I think it helps people understand a little bit when I've taught this before, is let's draw the heart in, okay. So you might be looking at this thinking, what the heck is going on here? Let's draw the heart in, okay. So here we're going to have our right atrium. Oh, that was a very rubbish drawing. So our right atrium is going to be here, okay? This one is going to be our left atrium. we're going to have our right ventricle. Let's just forget about the aorta and the pulmonary artery just for now, okay? And then let's draw our left ventricle, okay? So this part, this just helps you understand where we're looking at in the heart a little bit, okay? And it's important to know, I don't know if we've gone over it, guys, that there's 3 types of muscle in our body, okay? And there's only one type of muscle that we can control, okay? So we've got skeletal muscle, which is the type of muscle we can control, all right? So just think, you know, if you're moving your arm, you've got your biceps and triceps moving, working opposite pairs, okay? You've got your smooth muscle, which is found, you know, in your trachea. It constricts it. We've talked about it. It's in the arteries, okay? Helps to constrict and narrow and then widen, okay? And you can't control that, okay? And then you've also got your cardiac muscle, which, like it sounds on the box, is in your heart, okay? And again, you can't control that, okay? And what's interesting about the cardiac muscle is it initiates its own heartbeat, okay? So it doesn't need the brain telling it to beat, you know, if it's 60 beats per minute, so every second, it doesn't need to go, oh, you need to do a beat, you need to do a beat, you need to do a beat, okay? It does its own heartbeat, okay? And this is what you guys need to know about, all right? So there's a couple of things that are labeled on here that will, you know, come to light a little bit more. So let's start right at the top. So I'm gonna underline in pink so we can see here, okay? So our heartbeat comes from this node, which we find in our right atrium. So the top of our right atrium, we've got this sinoatrial node, all right, or SA. You can also call it the SA node, okay? All right. We've also got this other node called the atrioventricular node, or our AV node, okay? So an easy way I always remember which way around these two are is the AV node is between the atrium and the ventricles. So it's here, okay? Or S for superior, so at the top, okay? That's an easy way. You guys can come up with your own ways of remembering it, but that's how I normally remember it, okay? So what happens is our heartbeat comes from our SA node or our sinoatrial node. Okay, so like I said, it initiates its own heartbeat, so it's what we call myogenic. Okay, so what happens is a sinoatrial node sends out a wave, all right, and it depolarizes the right atrium. Okay, and that just means it makes it contract. It wants to make it contract. Okay, so let's draw here where our little impulse is going. So it's going all the way down here. Okay, so down here, you know, we've got this little one here. You don't need to know the names of these little fibers. Okay, so it's going to cause our right atrium to contract. Simultaneously, this electrical impulse is going down this fiber as well, and this one is called the Bachmann's bundle. Okay, and this is going down our left atrium. Okay, and this is going to cause our left atrium to contract. Okay, so both of them, we want them both to contract simultaneously. We want both sides of the heart to be doing pretty much the same thing. Okay, so we're starting off at our sinoatrial node. Okay, it sends out a wave, causes our right atrium to contract, sends down a wave down our Bachmann's bundle, and that causes our left atrium to contract. Okay. Now, what's important to know is between the atrium and the ventricle, we've got a layer, if I just draw it here, an insulating layer. Okay. And this doesn't conduct any electrical impulse or anything like that. Okay. So 1 second, sorry, I'm just moving James to another room. Perfect, there we go. So it means that the electrical impulse isn't conducted at the top of the ventricles there, okay? It doesn't conduct down. Okay, that's really important because we don't want the electrical impulse to conduct there. So let's do, if we didn't have that layer and the electrical impulses started carrying down the walls of the ventricle and started contracting, why do you think oh, that was a very rubbish arrow. Why do you think we don't want the ventricles to contract at the top? Any ideas? Why don't we want them to start contracting at the top? So a clue is we want them to contract from the bottom. Why do you think that is? Does anyone, any ideas? Okay, so Ria's put here, blood can't go through. So yeah, you're on the right lines there, Ria. So let's have a think. If we draw our heart at the side again, okay, let's just draw again our one side of the heart. So apologies for my very rubbish. So this is just our left side of the heart. Our atria contracts, sends blood down. Okay, if our ventricles start contracting at the top, they would push blood, they would start at the top and they would squeeze to the bottom and they would push blood down the bottom of the ventricle. Okay, we want blood to go out of the artery up here, okay, which is at the top of the ventricle, or yeah, anatomy, it looks easier. So we need that ventricle to start, you know, contracting from the bottom to push the blood up and out of that artery. So either the pulmonary artery or the aorta, depending on which side of the heart we're on. Okay, so we need the ventricle to contract from the bottom. So that's important. Okay, so like I said, how we do that is we've got this insulating fiber here that stops the electrical impulse traveling straight from the atrium to the ventricles. Okay, so that means the only place that the electrical impulse can go is to this node here, the AV node. Okay, and it gets held there for a second. Okay, so the AV node just holds it there for less than a second, just for 0.1 seconds or 0.2 seconds. Okay, and that's quite important, and that allows the atria to fully contract and all the blood to go from the atria to the ventricles. So it gives it a second to do that, or 0.1 seconds. Okay. Okay, great. So Ria's just put, what's the layer called? I'm not sure of the official layer. Don't need to know that, I don't think. I can double check for you, but it's just it's important to know it's just an insulating layer, okay, that doesn't conduct this electrical impulse. Okay. So great, so we've gone from the side atrial node down the right atrium, down the Bachmann bundle, and contracted the left and right atria. The blood's now in the ventricles, all right? So now we're at the atrioventricular node, okay? So we need to get that electrical impulse to the bottom of the ventricles. So between the ventricles, we've got the septum in the heart, and we've got this bundle, and it's called the bundle of His. Okay, so here they put His bundle, but I would call it the bundle of His. Okay, that's how I've always referred to it as. So the bundle of His. So it's a bit easier that way around. Okay, so that's our next bit. So that's where our electrical impulse is going to start going down. Okay, so then next it's going to go down and it's going to split off into either side here. Okay, and these are called the Purkinje fibers. All right, and how you spell it is on this, it's on this diagram here. Okay, Purkinje fibers. So it splits off into either side. Okay, and you can see they go all the way down to the bottom of the ventricle and go up, and then they start branching out. Can you see that, guys? And that means that the electrical impulse is taken right to the bottom of the ventricles before it starts, you know, allowing the ventricles to contract. Okay. Okay, great. And then that allows the ventricles to contract from the bottom, ventricular systole, and push all that blood up into either pulmonary artery or the aorta, okay, and push it right up. All right, so let's go through that again to make sure we understand. So this bit, guys, is just remembering the order. If you can remember it maybe in 5 steps, let's go through. So the SA node, or the sinoatrial node, sends out a wave of excitement. It causes the right atrium to contract, and it sends down the Bachmann's bundle and causes the left atrium to contract, okay? The electrical impulse can't travel directly from the atrium to the ventricles because of an insulating layer, okay? So the electrical impulse goes to the AV node, okay? The AV node holds the electrical impulse for 0.1 seconds to allow all the blood to enter the ventricles, okay? The AV node then sends the wave of excitement, or the impulse, down the bundle of His, which then separates off into the left and right Purkinje fibers, okay, right to the bottom of the ventricles, and the ventricles contract from the bottom up to send all the blood into the big artery we've got there. Okay. Any questions, guys? Again, we can go over it as well. So Ria, so here, what does the fibers do? It's just like a nerve, the fibers. Okay, so they just conduct the impulse down. They're just there to conduct the electrical impulse. Okay. So I will just learn maybe like 5 steps, okay, and that's what we do. Again, let's try and get a question next week so we can go through it. All right. So perfect. And any questions, you can just ask me and we can go back. Okay. So there's a couple more things you need to know. So we need to know about ECG, you need to know about hemoglobin, and then a little bit about the bore shift and things like that. So let's try and get through that. We've got 20 minutes, so plenty of time to go through. So the ECG, so let's have a look at this and what each part means, okay? So this is showing one beat of the heart, okay? So you might have 60 of these in a minute, and that means your heartbeat is 60 beats per minute, okay, which is perfectly healthy. So normal heart rates between about 60 and 100, but for your guys' age group, probably between 60 and 80 or so on. Or you can have it if you're quite fit and go to the gym or do sport, your heart rate can be low as like 40, okay? So it just depends. Okay, so let's have a look what each part of the ECG means. So you can see a few things here that we've got, okay? So you can see we've got parts labeled P, Q, R, S, and T, okay? So how we split this up is we say we've got a P wave, okay? We've got a Q, R, S wave, okay? And we've got a T wave, all right? So this P wave we get on this ECG, that's caused by atrial systole, okay? So the atria contracting, and that causes this little wave of excitement, okay? So remember at this point our SA node has just sent out that wave of excitement and caused the atria to contract. So atrial systole, okay? So remember the atria are a bit smaller than the ventricles, okay? So it only causes a slight wave, okay? Next, we've got our QRS wave, and this is ventricular systole. So this is the ventricle contracting, okay? Sending all that blood into the pulmonary artery and sending all that blood into the aorta, right and left side of the heart, okay? And it's much bigger because the ventricles are much bigger than the atrium, okay? And they've got, generally a little bit more muscle and more volume, okay? All right. And then we've got this T wave, and this is during ventricular diastole. Okay, so when the heart is at rest or nothing's pumping, that's what we're saying. Okay. So in your specification, there's 4 things you need to know that you could see maybe on an ECG. All right. So these are called, we'll go through the first 2. They're really easy. So tachycardia and bradycardia. Does anyone know what those terms mean? Anyone? I can see she shakes her head. Okay. So tachycardia. So I'll write these down. So tachycardia is when your heart is beating too fast. Okay. So you know how I said that your heartbeat should be between 60 and 100. So tachycardia is when your heart rate is over 100 beats per minute. Okay. So over 100. Okay. So then on the flip side, bradycardia is when your heart rate even, not your heartbeat, is less than 60 beats per minute. Okay. So less than 60. So I should put my units on here. So BPM, so beats per minute, that's all we write here. Oh. Okay, great. So in an exam question, I don't know, you might get given an ECG. Okay, so it will tell you an exam question. This ECG shows 10 seconds. Okay, so you might get a section of an ECG and it's 10 seconds. Okay, so you'll get let's say of these complexes, so these P, Q, R, S, and then T wave, you might get 6 of them. Okay, so if you get 6 of those in 10 seconds, going to want to know what's that person's heart rate. So they've got 6 beats in 10 seconds, so what's their heart rate? So, Salma, think in your head, does that make sense, that number? Remember, normal heart rate's between 60 and 100, okay? So if I've got 6 heartbeats in 10 seconds, in a minute, I need to get to 60, don't I? So I'm going to times 6 by 6, okay? So that's going to be 36 heartbeats in 1 minute, okay? So if we've got 6 in 10 seconds, that's going to be 36 in 1 minute. So that person's got bradycardia, haven't they? Their heart rate, is less than 60, okay? So again, if I've got my ECG and it shows that this person is having 20 heartbeats, separate heartbeats in 10 seconds, okay, their heart rate's going to be 120, isn't it? Yeah, perfect, Salma, thanks. 120. So they've got tachycardia, okay? And you can just see that on an ECG. So they might say, I don't know, Robin's heart rate is 130. What does Robin have, or what is the term for that? And you would say Robin has tachycardia. Okay, so that's too fast. So tachy, too fast, brady, too slow. Okay, I'm trying to think of a good way of remembering that. If anyone comes up with one, that'll be good. Okay, so perfect. So one more you need to know is an ectopic heartbeat. Okay, so does anyone know what an ectopic heartbeat is? And no worries if you don't, because these are probably new terms for you guys. So let's draw out a possible ECG. So let's do, so I've got on my P wave, my Q, R, S, and my T wave. Okay, let's go again. So my P wave, QRS, okay. So what ectopic heartbeat is can you see this little gap between my heartbeats here? Okay, so I've got a normal little gap. So what might happen is we might get ectopic heartbeat. So you get just a random heartbeat. Okay, so it's too early. Okay, so I might get a heartbeat that comes right here. Okay, but then I get my normal pause. QRS. T. Okay, so we can see this heartbeat came far too early. Okay, not right, should have been a little bit further along, shouldn't it? And that's what we call an ectopic heartbeat. All right, so just too early. All right, perfect. So bradycardia, tachycardia, ectopic heartbeat, and the last one you need to know is fibrillation. Okay, so fibrillation is just when you can't really see this PQRST waves. It's very random and all over the place. Okay, so there's a few different types of fibrillation that you guys don't need to know about. You just need to know that it's an irregular heartbeat. All right, so you know when people are shocked when they had CPR and they have an electric shock, their heart might be in fibrillation there, so we shock their heart to try and get it into that normal heartbeat. All right, so we might have a heartbeat that just kind of looks like this. Okay, might have a bit of a gap, and then we just got this. Okay, it might just look like that. Okay, so it's just, you can't make any sense of it. You can't tell what the heart rate is. You can't see those nice PQRS waves or the T wave. Okay, it's a little bit all over the place. All right. Again, next week we'll do an exam question lesson. Okay, and we'll get all questions on these. All right. So bradycardia, your heart rate is less than 60 beats per minute. Okay. Tachycardia, your heart rate is higher than 100 beats per minute. So if you guys just remember that your normal heart rate should be between 60 and 100, and anything outside that, that's either bradycardia or tachycardia. Okay. Your ectopic heartbeat, so that's this ECG at the top here, is when you get a heartbeat that comes too early. Okay. And then you get a little longer pause after it where it should have been. Okay, and then fibrillation, you can't tell your heart rate, you can't see nice PQRST waves. It's a little bit all over the place, if you like to say. Okay, so if you guys can feel your heartbeat, so your pulse here just below your thumb on your wrist, have a feel. So maybe after the lesson, you can see what you guys' heart rates are, okay? So it should be nice and steady. So it should have a regular rhythm, if you guys can feel. Okay, so if someone's got fibrillation, it might be beating really, really fast, and then it might have a pause, and then beat, 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 have a pause, and then maybe go to a normal heart rate and keep going, okay? So the rhythm is all over the place, all right? If it's an ectopic heartbeat, you might feel your pulse, and then you might feel it go boom, boom, really fast, and then a little pause, and then it starts again normally. Okay, so that's your different ways there. Great. So let's try and finish this topic off here. So again, any questions, just speak up or send me a message in the chat. That's great. Okay. Our next little bit in this topic, nearly at the end here, is hemoglobin. So we learned, I think it was last lesson, wasn't it, about, or the lesson before, about hemoglobin's found in red blood cells. So remember, red blood cells don't have a nucleus because they want to pack as hemoglobin in so they can pack as much oxygen in. Okay, so this is what hemoglobin looks like. All right, so we did a little bit ages ago about proteins and our primary structure of a protein, our secondary structure of a protein, our tertiary and our quaternary structure. Okay, so remember, our primary structure was just our sequence of amino acids. Okay, Our secondary structure had some bonds between them, okay? Our tertiary structure, it was all folded up. We've got loads and loads of different bonds, okay? And that might make an active site of an enzyme. And then our quaternary structure is lots of tertiary structures stuck together. So that's important because we can see here that hemoglobin is made up of 2 beta chains and 2 alpha chains, okay? So hemoglobin has a quaternary structure, okay? Because these beta chains and alpha chains, these are tertiary structures, okay? So we need to transport oxygen around the blood. And how we do this is it binds to hemoglobin, all right? So an important thing to know is each hemoglobin can take up to 4 oxygens, okay? So we have a little equation here that I will write for us. So 4 O2s plus hemoglobin, I'll just call heme, equals, well, I'll do a little arrow like we should do, oxyhemoglobin. Okay, so that's our little equation here. And there's the, this is what it looks like if we're doing here. So HbO8. Okay, so each hemoglobin can take up to 4 oxygens, okay? And it takes up to 4 oxygens in the lungs. So you remember our gas exchange across the alveoli to the capillaries, okay? That's where we want to get our oxygen from. And we want to get the maximum we can. So we want to get 4 oxygens. And as we travel around the body, so then we go into our pulmonary vein, remember, from the lungs, back into the heart, the left side of the heart, pumped up out of the heart, our aorta, and round the rest of the body, okay? We go into our arteries, our arterioles, capillaries, and then back into our venules and veins, okay? So we want to keep our oxygen in our hemoglobin until we get to where it's needed. So if you think, if you're running, okay, it needs to be in your legs or your arms. You need to get that for your respiring tissues, okay? They're in your legs and in your arms, so your arm and leg muscles, okay? So we want to try and keep it like that. All right, great. So we'll just conscious of the time, we've not got loads of time to go through this, so let's talk a little bit more about hemoglobin, and we need to know a little bit more of the equations. Let's move on to the oxygen dissociation curve, and then we'll come back to hemoglobin at the start of next lesson. Okay. Ooh, let me delete those annotations. So this is the Bohr shift, which we'll talk about in a second. But what happens is at levels of high oxygen, okay, or at a high partial pressure, if we can see it down at the bottom here, the saturation of hemoglobin with oxygen is 100%. So it's got those 4 molecules of oxygen, okay? And that's what we want, don't we? We want our oxygen to be in our hemoglobin as much as possible, okay? So here, partial pressure of oxygen, let's say 14, let's say we're at the lungs, all right? Now we've traveled to the left side of the heart to be pumped around the body. Let's say here, the partial pressure of oxygen is 12. So let's go down, all right? Just ignore this low PO2 curve just for now, because that's our next bit we'll talk about, okay? So the hemoglobin, you can see, loses a little bit of oxygen when it travels. So let's draw a straight line. So it was at 100%, now it's at, let's say, 97, all right? So we keep going down, now we're in the arteries, all right? So we're at 10 partial pressure, okay? Let's have a look what we've lost. So we're at, what, 96%. So we're still pretty good, aren't we? Gone to our arteries, now we're in our arterioles and we're going to our capillary bed, okay? So let's say that's at 8 partial pressure of oxygen, All right, so let's draw across again, so at 8. Oh, didn't mean to draw a square. So let's say that's at 92. All right, so we're still pretty good. Now we're getting to our muscles. So let's say we're in the arms because that's where it was needed. Okay, and let's say the partial pressure is 6 within our muscles. Let's go down. So we can see now that our oxygen in our hemoglobin is at 70%. So there's been quite a big drop there, hasn't there? But we need our oxygen in our muscles. So it's a great place for our hemoglobin to lose oxygen within our muscles. Okay, let's keep going. Let's say now our muscles are at 4. So let's go down at 4. Look at that. Our saturation is now what? 18%. We've lost loads of our oxygen in our muscles. But again, that's where we need to lose oxygen. All right. And as we go lower down, let's say that's about what, 6 there or something, 6 or 7% saturation. So we've lost all of our oxygen. Okay. So now that hemoglobin inside our red blood cells, inside our blood, is going to travel back in our venules and our veins to go back into the right side of the heart, to be pumped to our lungs, to be reoxygenated. All right. So the whole thing with hemoglobin is at high partial pressure of oxygen, it has a high percentage saturation of oxygen. Okay. Up until about 8, about here, it was about 90%, wasn't it? So it still kept a high amount of oxygen. So that's really important because we don't want to lose our oxygen when it's just traveling down in our arteries. We don't want to lose our oxygen. We need it to get to our muscles. All right. And then you can see here, it's really steep. Okay. So when it gets to quite low partial pressure of oxygen, so when it got to 6, we knew that it had gone down to 70%. So it lost about 20% just in that movement from 8 kilopascals to 6 kilopascals. Okay. So that means it readily dissociates when it gets to where it's needed. Okay. Again, as we go down to even less oxygen, partial pressure of oxygen, it goes down, what did we say this was, about 18. Okay. So it's lost 50% of its oxygen's hemoglobin, or 52%. We go down again, what do we say this was, about 6. So another 12% lost, but it's lost where it's needed. Okay. So at hemoglobin, it's adapted for its function because at high levels of oxygen, okay, it has a high percentage saturation of oxygen. Okay. So it binds with oxygen really well. As we start to go lower, it keeps hold of its oxygen quite well until it reaches a point and it starts to readily dissociate down. Okay. And it means it can transport oxygen really well. Okay. Because it can hold it till it's needed in the muscles. Okay. All right. So we've run out of time a little bit there, guys. So we'll talk about this Bohr shift and the high and low carbon dioxide, and we'll talk a little bit more about hemoglobin at the start of next lesson. Okay, so let me just stop this recording here.